Hi, this is Dr. Collins Jones. Welcome to segment 7, the final segment of my video series on manual cell counting and cell density calculations. In this segment, we're going to learn about calculations for cell seeding at specified concentrations, including dealing with anchorage-dependent cells where we have to seed at cells per centimeter squared, and batch seeding where we want to seed multiple flasks at the same time. Let's get started. Seeding flasks at a specified cell density. Cell seeding calculations are used to determine the volume of cells required to seed a new flask or flasks at a specified starting cell density. And this is important because, for example, if you're in biomanufacturing, the batch record may indicate that you have to seed the bioreactor at a specific cell density at a specific percent viability. So you have to know what volume of cells to put into that bioreactor to have a starting concentration that's specified in the batch record. Similarly, if we're going to do, say, cell-based assays to look at the potency of a new drug, we want to start in our multi-well plate. We want to start with the same concentration of cells in every well, so that the only variable is the drug that we're using or the concentration of the drug we're using. So it's important to be able to seed a flask or flasks at a specific starting concentration of cells. And that's what we're going to learn how to do in this segment. So for example here, I like to call the initial flask the source flask. So this is the flask that's going to supply the cells to seed the next flask, which is the destination flask. All right. And so here, what I've just arbitrarily made up some numbers here. The source flask is at a concentration of 6.30 times 10 to the 7th cells per mil, and our destination flask, we want to seed that at 5.00 times 10 to the 5th cells per mil. And I've also indicated that we want this to happen in a final volume of 40 mils. So what we're trying to calculate here is how many mils of cells do we need to take out of the source flask and put into the destination flask to achieve this concentration. And then we might need to go further, where now this initial destination flask becomes the source flask for our next destination, which could be a benchtop bioreactor, in this case a 3-liter benchtop bioreactor. And again, here we're going to start with however many cells are in this flask after they've grown for 5 to several days, and we want to then seed this bioreactor at a starting density of 1.5 times 10 to the 6th cells per mil in 2,500 mils or 2.5 liters. And again, we're just trying to see how many mils of this culture do we need to put into here. Okay. All right. So this is a dilution problem, right? You might have guessed that by saying we have a concentration here, and a volume here. Okay. So all dilution problems can be solved using the equation C1V1 equals C2V2. And just a quick review of this, right? C1 is the initial concentration, that is the highest cell density. This will be the concentration of the source flask. V1 is the initial volume. This is the volume of cells that we're going to passage from the source flask into the destination flask, right? So this will be the smaller volume. C2 is the final concentration. So this will be the concentration of cells that we want to obtain initially or starting in the destination flask. So C2 is the starting concentration of cells in the destination flask. So this will be a concentration that is smaller than the concentration in the source flask. And then V2 is the final volume, right? The combination of the volume from the source flask and the volume that we want to attain in the destination flask. V2 is the final volume in the destination flask. Okay, just an important reminder, C1 and C2 must have the exact same units, and when we do cell density calculations, those units will be cells per milliliter, and V1 and V2 must also have the exact same units, either mils or liters. So at a small scale, the units that we'll use will be milliliters, but if you start to proceed to a larger scale, then it'll be liters. And at some point in time, I will write another series of videos in dealing with bioreactors and how to do some of those calculations. So again, what we're going to be doing is calculating the volume of the seed culture, that is the volume of cells from the source flask that must be passaged into a final volume of media in the destination flask to achieve a specific starting cell density at a specific volume. So in order to do the C1V1 equals C2V2 calculation, we need to know the cell density of the source flask, right, which we're going to obtain by counting, either manual cell counting or automated cell counting, but that will give us the concentration of cells in our source flask. And then we need to know the concentration of cells in the destination flask and the volume of the destination flask. And more often than not, that will be specified in the protocol, in the SOP or in the batch record, but those will be indicated as part of the documentation. Let's start with some examples then. Example 1. Determine the volume of cells required to seed a 125 ml shake flask containing a total volume of 15 ml at a concentration of 2.00 times 10 to the 5th cells per ml, and describe the seeding process. The concentration of cells in the source flask is 5.84 times 10 to the 6th cells per ml. Okay, so as always, I kind of like to just diagram out what it is we're doing. And so I just draw a quick little picture here. We have 5.84 times 10 to the 6th cells per mil, and this is our source flask. We have 2.00 times 10 to the 5th cells per mil, and we want that to be in a 15 mil volume. And so this is our destination flask. So this is indeed a C1V1 equals C2V2 problem. So we'll start by identifying C1, C2, V2, and proceed from there. Okay, I know I skipped one, sorry. But you guys get the idea. So C1, we have two cell densities, 5.84 times 10 to the 6th, 2.00 times 10 to the 5th. So the larger cell density, that concentration is going to be C1. That means the other concentration, C2, the smaller concentration, is going to be 2.00 times 10 to the 5th. So remember, C1 is always the greater concentration value. V1 is what we're trying to find. Remember, this is the volume of cells in the source flask that we need to passage to the destination flask. And V2 is the volume that we want to end up with in the destination flask. In this case, it is 15 mils. Okay, so V2 is always the volume we're preparing, and it's going to be the larger volume. So let's proceed with the calculation. Again, C1V1 equals C2V2. Right, we're just going to plug the numerical values into the equation, and then we're going to solve for V1. Okay, and so when you do that on your calculator, here I put down all of the digits on the display, but remember that when we're dealing with cell culture, we're only going to go 2 places past the decimal point for significant figures. So I'm going to need to transfer 0.51 mils of cells from our source flask into the destination flask. So just practically thinking, what would you use to do that? In my lab, I would either use an autoclavable P1000 and of course autoclaved tips, or I would use a sterile 1 mil serological pipette to do the transfer. Okay. Now, one other thing that we need to think about then is how much media are we going to add, right? Because we want a final volume of 15 mils. So the mils of media to add would be the total final volume minus the volume of source cells. So in this case, this would be 15 mils, right? The volume that we wanted here, total volume of 15 mils minus 0.51 mils, all right, or 14.5 mils. I know it's 14.49, but we would probably in the real world round this up to 14.5. Okay, so seeding process. First, observe good documentation practices. So record traceability for all the critical to quality attributes and any other information that's specified in the SOP or the batch record. And then we will add 0.51 mils of cells that are 5.84 times 10 to the 6th cells per mil into 14.5 mils of media. And so remember, we always add media first, right? So we'll add the media into the flask, and then we'll transfer 0.51 mils of cells from the source flask into the destination flask. By adding media first, it ensures a better dispersal of the cells from the source flask. They distribute themselves more uniformly if we add the cells into the media and then give it a little swirl or a little shake. And at the very end of this video, we'll describe some mixing patterns and tell you what is supposed to be the best. Okay, next problem. So I want you to try this, and I'm going to count to 10 so you can pause the video and then read the problem and try to work this out on paper, and then come back and see how your answer, your solution compares to mine. All right, I'm back. Let's see how you did. So first, and of course you don't have to do this, but I really do like to just make a sketch of the process so I can keep track of what volumes, what concentrations go where, and just have a picture in my head of what's going on here. So again, we have a T75 flask containing 14 mils of media, and the density of cells in there is 4.80 times 10 to the 6 cells per mil. So this is the source flask, and we want to seed a T225 flask at a final volume of 38 mils with a cell density of 5.00 times 10 to the 5th cells per mil. And again, this is our destination flask. So here we are. A C1V1 equals C2V2. So again, we want to identify C1, C2, V1, and V2. We want to just match the numbers to each of those variables. And even though this takes a little more time, I like to do it like this because it's easier to look back if there's an error in the calculation. It's easier to figure out where you made your mistake. Did you put the wrong number in the wrong place? It just helps things to go linearly. It's easier for other people to audit your work, and by doing things in a specific pattern, you're less likely to make a mistake. So C1, right, the concentration of the source flask is 4.80 times 10 to the 6 cells per mil. C2 is the concentration we want to end up with in the destination flask, 5.00 times 10 to the 5th cells per mil. V2, that's the volume of source cells that we want to passage into the destination flask. So that's what we need to calculate. And then V2 is the final volume that we want to end up with in the destination flask, and that is 38.0 mils. All right, now notice, right, that the 14.0 mils that I mentioned in the source flask is irrelevant to this calculation. It's just extra information. We don't need it for the calculation. It may, however, that volume may, however, be important to consider when we actually do the seeding, okay? So for now, we can just ignore that volume. All right. So let's proceed with the calculation. C1V1 equals C2V2. Let's plug the values into the equation. Solve for V1. Again, I just printed out the display on the calculator, but it's 3.95833. So we're going to round that up to 4.0 mils of cells. Again, one place past the decimal point because here we're probably going to use a 5 mil serological pipette for the transfer. Okay, so we know how many cells we need to take out of the source flask and passage into the destination flask, but now we also need to think about the media. So the mils of media is going to be the total final volume minus the source cells volume. So in this case, it's going to be 38.0 minus 4.0, or 34.0 mils of media. So the seeding process, again, adhere to good documentation practices, record traceability for all the critical to quality attributes, as well as any other information as specified by the SOP or batch record. And so now we're going to add 4 mils of cells that are 4.80 times 10 to the 6 cells per mil into 34 mils of media. So again, we put the media in the flask first, and then we passage the 4 mils of cells into that 34 mils of media. And then we disperse this by moving back and forth and side to side in linear motions. And again, adding the cells into media that's already in the flask gives us a better, more uniform dispersion of cells throughout the flask. Let's try another one. Okay, so again, you all can read this. If you want, you can sketch out the process and then go ahead and solve the problem. So I'm going to count to 10 again and then come back and let's see how you do. All right, I'm back. Here we go. Again, I just like to sketch out the information in the problem. So we have our source flask. It's 1.37 times 10 to the 6 cells per mil, and we have a volume of 25 mils. We want to then passage this into a shake flask, right, at 7.5 times 10 to the 5th cells per mil, final volume of 75.0 mils. So this is our destination flask. Proceed with the calculation. Hey, no surprise here. A C1V1 equals C2V2 problem. So again, identify C1, C2, V1, and V2, right? So let's just write that out, and I'm abbreviating a little bit here because by now you should be getting comfortable with this. So once again, we've identified C1, that's the larger concentration, and that's going to be the concentration in the source flask. V1 is what we're trying to find. That's the volume of cells we need to passage from the source flask into the destination flask. C2 is the initial or the starting concentration of cells in the destination flask. And 75 mils is the total volume that we want in the destination flask. Now, once again, note the 25 mils that I gave you as the volume in the T150 source flask. At the moment, this is just extra information not needed for the calculation, so we're going to ignore it. Again, this volume may become important when we talk about the actual seeding process. So let's go ahead and do the calculation then. Okay, step 2 here is plug and chug. So just put the values into their corresponding positions in the equation, solve for V1, and as we run these numbers through our calculator, the answer is 41.05841 or 41.4 mils. So that's the volume of cells that we need to remove from the source flask and put into the destination flask in order to achieve a starting concentration of 7.50 times 10 to the 5th cells in a 75 mil volume. All right, but wait, right? We see this number here, right? And that's the number that we're supposed to transfer. But look, there's only 25 mils in the source flask. So this is not good. We are not going to be able to do this process. We cannot carry out this procedure because the T154 source flask only has 25 mils of total volume. So seeding the destination flask at the specified volume and to get the specified cell density cannot be done with this particular culture. So either we need to have a flask with a greater volume of cells at 1.37 times 10 to the 6 cells per mil, or we need to grow the cells to a higher density in the source flask. But either way, we are not going to be able to meet the specifications indicated here, so we cannot proceed. And of course, that also needs to be documented, right? All right, and it'll be a deviation. And now we need to go figure out why we didn't have enough cells and enough volume, and if we need to do a corrective and preventative action so that this doesn't happen again. All right, example 4, here you go. Right, let you read this and then let you solve the problem. And again, I'm going to sit here and count to 10 and then come back and we'll see how we do. All right, let's go. Once again, diagram out the process, right? So we have 2.38 times 10 to the 7 cells per mil and 60 mils of media in the source flask. And our destination flask, we're going to have 7.25 times 10 to the 5th cells per mil. That's the starting concentration we want in a volume of 350 mils, and this is our destination flask. And obviously I didn't try to draw this to scale, but it sort of shows what numbers go where and what we are doing. So this still works for me. Hey, another C1V1 equals C2V2 problem. So once again, assign values to C1 and C2, and V1 is what we're trying to find. That's the volume of the source flask cells that we need to passage into the destination flask. And V2 in this case is 350 mils. Okay, once again, the volume of cells, the 60 mils in the source flask, is not needed for this calculation, so we can ignore it. Solve the problem, C1V1 equals C2V2. We're just going to plug each value in to the corresponding variable in the equation. Solve for V1. And again, when we solve for V1, after the round off, we see that we have 10.7 mils of cells. All right. Okay. But now we also want to calculate the volume of media that's required. So the media volume is going to be 350 mils, the total volume we want in the destination flask, minus the volume of cells from the source flask that we're going to passage. So this is 339.3 mils. Oops, and I left the unit off. That's not good. That will be a ding on an audit. Okay, so make sure that you put units with all of your calculations. That's my mistake. So again, we're going to add 339.3 mils into the destination flask first, and then we're going to passage 10.7 mils of a cell culture that is 2.38 times 10 to the 7th cells per mil. We're going to passage 10.7 mils of that cell density into our destination flask, and then give it a little swirl and put it on the shaker. Now, this is sort of a new one, right? A little bit different. Here, we're going to talk about seeding densities by surface area, that is centimeters squared. So this applies to anchorage dependent cells when we're seeding T-flasks, dishes, hyperstacks, and things like that. So here's the information we're given. A cell culture is at a concentration of 2.50 times 10 to the 6 cells per mil. Determine the volume of this culture needed to seed a T225 flask at 1.00 times 10 to the 5th cells per centimeter squared. So we did not give a final volume because we're actually seeding by surface area, but we do have to make sure at the end of the day that there's enough volume in there to feed the cells. Right? So again, let's diagram this out. Here's our source flask. Concentration is 2.50 times 10 to the 6 cells per mil. And again, just a reminder, we obtained this value by either manual or automated cell counting, right? So we had to remove some cells, count them, and determine the concentration. And now in the destination flask, we're talking about how many cells per square centimeter on the surface, because these cells are going to be adherent cells. They're going to attach to the surface, right? And if they are normal cells, they will exhibit contact inhibition. They won't keep piling up. So once this flask is confluent, the entire surface is covered with cells. They'll stop dividing. So we want to start at a specific concentration, that is a specific number of cells per centimeter squared. That's the twist on this. All right. So this is really going to just become, if you will, a dimensional analysis problem. Very similar to the types of problems that you did in beginning chemistry or beginning physics or whatever. Okay, so first thing we're going to do is calculate how many cells we actually need. So the total number of cells needed is equal to the cells per centimeter squared. That's our starting concentration of cells, if you will. That's the starting amount of cells times the flask surface area. So how do we know the flask surface area? That's given in the name of the flask. So T225, that's 225 centimeters squared. A T75, the surface area is 75 centimeters squared. A T25, it is 25 centimeters squared. So the packaging on the flask will tell you the square centimeters. All right, so now we just need to plug this in. We want 1.00 times 10 to the fifth cells per 1 centimeter squared, and we need to cover 225 centimeters squared. So this is just a simple plug and chug, right? And so this is 2.25 times 10 to the 7th cells, because centimeters squared cancels out. And some people may do this as one continuous problem. I broke it up into two separate parts because I think it's easier to understand what's going on here. So now that we know how many cells we need to calculate the volume of the source culture that will contain 2.25 times 10 to the 7th cells. So again, we're back to how many mils do we need. But this is not going to be a C1V1 equals C2V2 problem. It is not going to be a C1V1 equals C2V2 problem because we don't have the third number. We don't have the final volume here, right? We just have a starting density and we have a cells per centimeter squared amount, all right? So now what we're going to do is we're going to treat the cell density, that is the number of cells per 1 mil, we're going to treat that as a conversion factor. So the volume of cells required will be the total number of cells needed that we calculated in step 1 times 1 mil over the number of cells. So this is just the cell density inverted, right? We've just switched it around because we're treating this like a conversion factor. 1 mil equals, in this case, 2.50 times 10 to the 6th cells. That's really what that means, at least for the purposes of this problem. So the volume of cells required is going to be 2.25 times 10 to the 7th cells times 1 mil divided by 2.50 times 10 to the 6th cells. And so again, you can see we've just inverted the cell density. Okay. And so the volume of cells required is 9.0 mils of source cells. All right. So we need 9 mils of cells from here. When we put this into the flask and spread these out uniformly, that will correspond to 1.00 times 10 to the 5th cells per centimeter squared. Now, we didn't address the fact that we need media, right? And 9 mils of cells is not going to be enough media for those cells to survive in a T225 flask, right? It won't even cover the surface of the entire flask, and the media will dry out and the cells will die. So it turns out that most cells grow best when there's between 0.2 to 0.5 mils of medium per centimeter squared. Okay, so someplace between 0.2 mils and 0.5 mils of media per centimeter squared is the optimal amount. And you would have to determine whether it's 0.2 or 0.3 or 0.4 or 0.35 or whatever mils per centimeter squared experimentally. That's what process development might do for you. So for us, I'm going to say that we have 225 centimeters squared. That's the size of our T225 flask. And I arbitrarily said, let's make it 4 mils of media per 1 centimeter squared. I could have just as easily said 0.2 or 0.3 or 0.5. I just chose 0.4 mils of media per 1 centimeter squared. So when we do the multiplication, we have 90 mil total volume in the flask. So the actual amount of media will then be the total volume minus the 9.0 mils of source cells, or 81 mils of media. All right. And so once again, what we're going to do is add 81 mils of media into the T225 flask, and then we're going to add 9 mils of cells from the source flask. We're going to make sure that those cells get uniformly dispersed throughout the flask, and our starting density will then be 1.00 times 10 to the 5th cells per centimeter squared. Okay, so not too difficult really, just need to pay attention to your units. All right, let's try this again, okay? An SOP states that a T225 flask should be seated at a density of 5.00 times 10 to the 4th cells per centimeter squared. The density of the cell culture used for seeding is 6.25 times 10 to the 6th cells per mil. What volume of source culture is required for seeding, and what volume of media might be appropriate for this. So I didn't write that part in, but we're always thinking if we're passaging cells, we have the cells that we're passaging and the media that's required to feed the cells. Okay, so once again, diagram out the process. 6.25 times 10 to the 6 cells per mil in our source flask. 5.00 times 10 to the 4th cells per centimeter squared in our destination flask, right? Again, no final volume is given. So if we don't have two concentrations in a volume, then this is not a C1V1 equals C2V2 kind of problem, right? So we're just going to do this the same way before. 1, calculate how many cells are needed. So the total number of cells needed is the cells per centimeter squared times the surface area of the flask. Again, this is a T225, right? And we want 5.00 times 10 to the 4th cells per centimeter squared, or 1.125 times 10 to the 7th cells. And again, you notice here, right? And some of this I'm doing deliberately, or at least pretending that I did it deliberately. You notice here that I have too many significant figures, right? We really only want to go 2 places past the decimal, but I could justify that by saying we don't round off until we've completed the entire calculation. So maybe I could get away with that. All right, now we need to determine the volume of source culture that will contain the required number of cells. So just like before, we're trying to figure out how many mils of the 6.25 times 10 to the 6 cells per mil do we need to passage into our destination flask. And again, it's going to be the same thing. The volume of cells required is going to be the total number of cells needed times 1 mil over the number of cells. And again, this is just an inverted cell density. So we just plug the numbers in. 1.125 times 10 to the 7th cells that we calculated as step 1 times 1.00 mil per cell, 1.00 mils per 6.25 times 10 to the 6th cell. So we've just, again, inverted the cell density that we obtained when we counted cells from this flask. If we do the math, it turns out that we need 1.80 mils of source cells. And here I have followed the correct number of significant figures for my final answer. And now, of course, I also need to determine how much media to add. So again, same thing, right? This is a rule of thumb, right? If you don't know how much media you need to end up with, this is a general way to calculate it, between 0.2 to 0.5 mils of media per centimeter squared. And so in this case, I just selected 0.2 mils of media, right? So we're going to multiply the surface area, 225 centimeters squared, times 0.2 mils of media per 1 centimeter squared. And again, this number, I just chose it arbitrarily, but partly I chose a smaller number to just show you how much it decreases the volume. So here we need 45 mil total volume. So the volume of media to add will be 45 mils minus the volume of source cells, 1.80, or 43.2 mils of media. Okay. And then again, we add the media into the flask first, and then we add the source cells, and then we move this linearly and from back and forth and side to side in a linear fashion to uniformly disperse the cells. And of course, you notice too, I left off the good documentation practices about recording the traceability, that is, the critical to quality attributes and any other information as specified by the SOP or batch record. So hopefully with these two examples, you understand how to calculate a seeding amount when we're going by centimeters squared, while we are going by surface area. Let's turn our attention now to seeding multiple flasks at the same time. So in this example, describe how you would seed 4 125 ml shake flasks, each at a final volume of 30 mils and a starting cell concentration of 6.50 times 10 to the 5th cells per mil. The concentration of cells used for seeding, that is the source flask, is 8.26 times 10 to the 6th cells per mil. So again, just to sort of picture the problem, to diagram it out, this is a scale out problem. All right, not a scale up where we're increasing the volume. The volumes may stay the same, but we're increasing the number of flasks. So here's our source flask at 8.26 times 10 to the 6 cells per mil. And now from this one flask, we want to generate 4 destination flasks, each of which has 30 mils of media in it and is at a starting concentration of 6.50 times 10 to the 5th cells per mil. So there are two things we need to think about here, right? This is a C1 V1 equals C2 V2 dilution problem. And we're just going to do the same dilution 4 times, once for each flask. This is one way to do it. On the next slide, I'll show you a different way to do it, right? And so whichever gives you the most reproducible seeding densities, starting densities, that's the method that you choose. So for this one, I might choose this method. All right, so step 1, as always, identify C1, V1, C2, and V2. And V1, once again, is what we're trying to find. So plug and chug here, right? So C1 is 8.26 times 10 to the 6 cells per mil. We're trying to calculate V1. That is, what volume of the source flask do we need to transfer into each of the 4 new destination flasks. And then each destination flask, C2, is 6.50 times 10 to the 5th cells per mil. And each destination flask will have a final volume of 30 mils. So it turns out that we want to do 2.4 mils from our source flask into each of the destination flasks. Right? And again, we need to calculate the media. And so the media is 30 mils minus 2.4 mils, right? Final volume minus the volume of the source cells, or 27.6 mils. So what this means is we would add 27.6 mils of media into each of these flasks, and then we would take 2.4 mils out of this flask and add 2.4 mils into each flask. And now all 4 flasks have been seeded at the same starting density and the same final volume. Okay? All right. So one challenge with that is we're having to open and close these flasks multiple times. If we have good aseptic technique inside the BSC, this should be okay, but this is one way to do it. Another way to do it is to do this from a batch culture. So here we're saying prepare 4 125 ml shake flasks, each containing 20 mils of cells at a starting concentration of 2.00 times 10 to the 5th cells per mil. The source culture is 50 mils of cells at 6.29 times 10 to the 6th cells per mil. And I want you to do this now from a batch culture. And so we'll see what that means. But one flask is going to serve as the source for each of the destination flasks. And so let's sort of see how that's done. Okay, so there's our source flask. 6.29 times 10 to the 6th cells per mil, 50 mil volume, and our source flask. So the question is, I want you to prepare a batch solution. And I'm saying 100 mils of batch solution at 2.00 times 10 to the 5th cells per mil. Well, how did I arrive at 100 mils, okay, in that batch flask? The volume of the batch to prepare will be the mils of batch is going to be equal to the total number of flasks, which is 4, times the mils per flask. So we have 4 flasks times 20 mils per flask, which is 80 mils. But I'm going to round this up to 100 mils arbitrarily. I don't need all of that, but 100 mils is just a nice, easy number to work with. All right. And the reason for that is if I made exactly 80 mils, I could not remove 80 mils from the batch flask. Some droplets always stay behind. So we always need to add more volume than we're going to remove. And I like to add in just an extra volume or two in case there's a mistake. All right. So I've just decided that I'm going to prepare 100 mils of batch flask. You could have done 90, you could have done 110, you could have done 120. You know, part of how much extra you prepare depends on how much everything costs. If the media is really expensive, I might only prepare an extra 2 or 5 mils. If the media is cheap, I might go to 100 or even 120 mils. Right? So those are the practical considerations that you need to take into account. So what we want to do now is using this 6.29 times 10 to the 6 cells per mil source flask. We want to prepare 100 mils of cells at 2.00 times 10 to the 5th cells per mil. So guess what? C1V1 equals C2V2 problem, right? Nothing changes. So again, we identify the values for C1, V1, C2, and V2 based on the information we were given in the problem. Okay? And so we just plug that into our C1V1 equals C2V2 equation, solve for V1, and it turns out that we need 3.2 mils of source cells. That is, we need 3.2 mils of 6.29 times 10 to the 6 cells per mil. The media volume that we need then will be 100 mils. That's the volume of batch solution that we wanted to prepare, minus the 3.2 mils of source cells gives us 96.8 mils. So what we're going to do here is combine 96.8 mils of media, that goes into the flask first, and then add in 3.2 mils of the source culture. All right. And now what we do is we take 20 mils of suspension from the batch flask, because those 20 mils will all be at 2.00 times 10 to the 5th cells per mil, and we just pipette that into each of our destination flasks. All right. So we've made one large batch of cells at 2.00 times 10 to the 5th, and then we're just going to take 20 mil aliquots out of that and pipette it into our different destination flasks. All right, now the important thing here is that when you're doing these multiple kinds of passages, right, from the same flask, you need to make sure that you're constantly agitating the flask gently so you don't generate shear, but you need to constantly agitate the flask so that the cells don't settle, right? So you need to make sure that you have a uniform distribution of cells throughout the flask, side to side, top to bottom, analytically identical throughout, all right? And that means that you have to mix frequently. All right, seeding density calculations for multi-well plates. So here's a multi-well destination plate, and what we want here is 2.50 times 10 to the 4th cells per mil per well, and we want a final volume in each well of 1.5 mils. So the idea here is that this is a 12 well plate, and so what we might have here is this might be an assay to test concentration efficacy for a drug, and so here we might have control cells, right, untreated cells, and then we may have different doses of the drug in each of these wells. And remember, if you've looked at my lab math videos in the statistics part, we always like to do things in triplicate. So we'd have 3 different doses here. 0 drug, dose number 1, dose number 2, dose number 3. And the only thing that we want to change is the drug, right? We want each well to have exactly the same number of cells so that the effects we see are due to the drug, not due to the number of cells per well. So I've given you a source flask here that has 8.28 times 10 to the 6 cells per mil. Okay. And so what kind of cells could these be? These could be cancer cells. These could be diseased cancer cells, or they could be even model cancer cells that we've purchased from the ATCC or from Lonza or been given to us by an academic research institution. But whatever, we've grown up these cells. We're confident that they will propagate and we can reproducibly grow the cells. So we have a source flask of cells at 8.28 times 10 to the 6 cells per mil. And so now how do we calculate how to get these cells into this well at this concentration in this volume? And for this, really the easiest, most reproducible way to do this is to do this as a batch. So first, we'll determine the total volume of media required for all the plates. So this would be 4 plates times 12 wells per plate times 1.5 mils per well, right? Said 4 12-well plates. Each well has a final volume of 1.5 mils. So again, this is really just a conversion, right? So if we do the math here, this turns out to be 72 mils total, all right? That's the minimum amount that we require. But if you recall from the previous slides, we said that you're never going to get all 72 mils out of your preparation. So we're going to make some extra. So for me, I decided that we're going to increase the volume in case a mistake happens and to have a little bit extra. So for me, it was easy to just go up to 80 mils. I arbitrarily chose 80 mils. You could have done 75 mils. Maybe you went all the way up to 100 mils. It doesn't matter as long as you do the calculation, the rest of the calculations correctly. But 80 seemed reasonable to me. Okay. All right. So we know our final volume is 80 mils. So now we're going to calculate the total concentration of cells required to seed 80 mils at a concentration of 2.50 times 10 to the 4 cells per mil. Guess what kind of problem this is? Yes, a C1V1 equals C2V2. So once again, we're going to identify C1, C2, V1, and V2. We're going to assign the respective values to each variable, plug that into the equation, and solve for V1. And V1 is 0.24 mils. Okay, so we're going to require 0.24 mils from our source flask, where the cells are at a concentration of 8.28 times 10 to the 6 cells per mil. And we're going to bring that into a final volume of 80 mils. So we're going to seed the plates, and what I would do is prepare 80 mils of batch, right, in a big flask, just like in the preceding slide, by adding 0.24 mils of the cells at 8.28 times 10 to the 6 cells per mil into 79.76 mils of media, right? That's 80 minus 0.24. And then in that batch, after I've mixed it, I would just remove 1.5 mils from the batch and add 1.5 mils of those cells at 2.50 times 10 to the 4th into each well. I would just disperse it. And again, be sure to agitate the batch flasks frequently to avoid settling. And once you've pipetted all the cells into here, right, being careful to avoid shear, so you're going to pipette slowly and evenly, then you would give the flask back and forth and side to side, right, front to back, back and forth, side to side agitation to make sure that the cells get uniformly dispersed throughout the wells. Okay, so final slide in this series. You made it, okay? Practical cell seeding. Can't emphasize enough. Add media to the flask first, then add the cells, or add the total volume all at once from a master batch at the correct concentration. This ensures a much better dispersion of the cells throughout the flask, especially if they are anchorage dependent cells. All right, and you need to work quickly because cells can settle in just a few minutes. And you can see here, after about 5 or so minutes, right, the cells have just begun to settle out of solution into a cell pellet. And so that means that you need to remix, right? And to do that, you'll tap it with your fingers or something to sort of agitate this, but you're not going to create a vortex. You don't want to create excessive shear forces because that will kill the cells. So we won't want to resuspend before seeding. Air bubbles can interfere with attachment, right? So this is especially a problem in multi-well plates. The smaller the wells, the more problems you have with air bubbles. And the other thing is that when air bubbles burst, they will kill cells. So if air bubbles are present and then you put your cells in and the cells are on top of the bubble literally explodes and it blows up the cells, right? And this is covered in a little more detail in the manufacturing class. Okay, so there are lots of different mixing patterns. The best mixing pattern really is when we do, front to back like this and side to side. So linear motion, nice and even like this, not too fast. Hopefully you can see my cursor here. Not too fast, back and forth, and then side to side, right? None of the circular stuff because you kind of get swirls and the cells may actually grow in that pattern, right? And no mixing at all. The cells just kind of stay where you put them. So if we pipette the cells into the media and then mix back and forth, side to side, we usually get a pretty good distribution. And that is the end of segment 7 and the end of my video series on cell counting and cell density calculations. Hope you found it helpful. Bye.